Right. Good afternoon, everyone. So welcome to the Afternoon Bioscience Day Symposium. Delighted to see all of you here. I'm Wolfgang Losert, the Associate Dean in the College of Computer, Math, and Natural Sciences. And on behalf of Dean Banavar, welcome you all. We're delighted to have a series of exciting talks on a topic that's really just like the college. So the College of Computer, Math, and Natural Sciences is a young college, 5 years old, and it was formed by connecting the physical sciences, computer and mathematical sciences with the life sciences. And the topic of today's symposium connects the physical sciences, computer sciences, mathematical sciences with the life sciences and with engineering. The topic is systems biology, advancing life science and medicine. And the first speaker, Marc Salit, is a leader in the Genome-Scale measurement group within the National Institutes of Standards and Technology. And at the same time, he's a consulting professor at Stanford University School of Medicine, and as well as in the Department of Bioengineering at Stanford. And so Marc has worked extensively on measurement science and chemistry and physics with an emphasis on precision measurements, lab automation, algorithm development. And his research is now focused on bringing this experience from chemical metrology to the emerging biometrology community. And he's the co-director of the Joint Initiative for Metrology and Biology, which is a partnership between Stanford, NIST, and industry, and leading a multidisciplinary team to establish measurement science. Welcome, Marc.
Well, thanks so much. Good afternoon, and thanks so much for inviting me across, Wolfgang, and thanks so much to the University of Maryland. I'm delighted to be back here. Some of the highlights of my professional life actually were back in 2010 and 2012 teaching a course that addressed the fundamentals. This one's still on. I'm going to turn that on. Fundamentals of metrology in chemistry and biochemistry. And from the work that I did with my colleague Dave Dewar here at University of Maryland, we really formalized some of our thinking around chemical metrology that ultimately led to some of the applications I'll talk about today in genome-scale measurements. It's delightful, as I say, to be here. NIST, actually, where I have spent the lion's part of my scientific career, is proximal. We're over in Gaithersburg, Maryland, headquartered. Oh, I'll show you that map in a minute. There's got to be a pointer here. With a significant campus, a significant asset with a couple thousand employees on site there. It's a national institute that is part of the U.S. Department of Commerce. I just wanted to give you guys a little bit of a context for this. There's a long partnership between University of Maryland and NIST, and in fact on site here, or maybe not on site, but a partnership laboratory, the Joint Quantum Institute, must be hitting its 10th anniversary right now, which is a joint institute between NIST and University of Maryland, hosted largely in probably the physics department here, but really taking some of the groundbreaking measurement science work done in the physics labs at NIST and bringing it towards practical application in quantum computing. And I bet I'm the only one that said practical application in quantum computing, maybe ever. We've got a partner lab that's been a long partnership also with first the University of Maryland Biotechnology Institute, UMBI, but now with the University of Maryland, and that's our IBBR, Institute for IBBR, Biomolecular Science. And that's over in, at the Shady Grove campus over in Rockville. Hollings Marine Lab is another key asset in NIST's portfolio where we do lots of measurement science in partnership with NOAA, the South Carolina Department of Natural Resources, the College of Charleston, and the University of MUSC, the Medical University of South Carolina. And an emerging cybersecurity institute, which is probably of less interest to this group right here, that's also located over there on the Shady Grove campus at this point. Another asset in Boulder, Colorado. You can read the sort of mission or vision for NIST. I'm part of the Material Measurement Laboratory. We're honored to have Lori Locascio, the director of that laboratory, with us this afternoon, whose focus is on delivering in the chemical, biological, and material sciences. And you can sort of see that list of portfolio highlights there. So just, I wanted to give context and I wanted to show the map because I don't live here anymore. I was on the Gaithersburg campus for 23 years and recently have moved out to the Bay Area in California to partner with Stanford University and Silicon Valley commercial sector, really Bay Area commercial sector in biosciences, and in particular in the genomics measurement science, really what we're calling genome-scale measurement science there. But you can see we've got sites in the material measurement lab at 8 locations around the country, trying to do what we had talked about. So what is this joint initiative in metrology for biology? I'm sorry, joint initiative for metrology in biology. We're just forming it up. We're working together to build the infrastructure so that biology can be reproducible, reliable, comparable, and scalable. And that scalability really comes through standardization and realization of distribution of labor. Let me use what you've done and build on it in a credible and reliable way. Right now we're formed up really with sort of 4 legs of a square here. There's faculty groups doing science and technology development, innovation. NIST trying to bring metrology in, as Wolfgang's introduction of me pointed out, trying to take what NIST have done so well in really, this is my elevator pitch is, what we're trying to do is do for the 21st century of biology what NBS and NIST did for the 20th century of physics. So I'm sure everybody's got a smartphone in their pockets or pocketbooks or rucksacks here, and that smartphone is built on the measurement science, really, that was done up in Gaithersburg and in Boulder over the last 100 years. And what is that going to look like 100 years from now when we're talking about biology? So we're trying to be ready for that. Not only ready for that, we're trying to create it. And so that's why we're out here trying to build this. Build that metrology, build a standards realization platform that can deliver on what we've been talking about or what I've been talking about. Do the measurement science. We're training the next generation, building workforce, and working closely with commercial partners to make sure that the stuff we do is actually the stuff that's needed to move the needle on bringing biology and biotechnology into commercial practice. And I wanted to give this context just to give the group a sense of the long timeline that NIST is considering and thinking about, as well as to give you a sense of my perspective and why we're doing the genome scale measurements that we are doing. This is some of the principles that are guiding our work in building this, and I'm working really closely with NIST headquarters as well as with faculty groups at Stanford to try and form this up. And at this point, we're using a small-ish set of consultants around the biotechnology space. And I say smallish, probably tens of different companies represented of the 1,000 Bay Area biotechnology and bioscience-related, biomedical related companies that you can pull up in a pretty easy Google search. So the upside potential is terrific for what we're doing. And again, what we're trying to do is provide the underpinnings to advance the science, engineering, and medicine applications of biology. I'm enough of an ad here. So who NIST are today, again, I wanted to come back to the metrology piece because I'm going to talk about forming up metrology in genome scale measurements. This is a traditional NIST NBS. NBS was the National Bureau of Standards, and NIST grew out of the National Bureau of Standards, NIST being the National Institute of Standards and Technology, grew out of NBS in 1988. And a recent moniker we're kind of, I think, enjoying is being called Industry's National Laboratory. And it's a great place to be, coming from the Department of Commerce, to have our customers recognize us as a resource and an asset. There's that story. So what is metrology? It's not telling you it's raining outside. It's the science of measurement. And what is that? Why do I care about that? And how do I do that? And so really what metrology is the stuff that goes around a measurement result that lets you know how to use it, that lets you know how to compare it to another measurement result, that lets you know how well to rely on it. Is this number different than that number? That lets you understand when your scientific experiment is seeing an artifact or when it's seeing a signal that in fact is going to lead you to your next set of experiments and lead you to reliable refutation or support of the hypothesis you're trying to test. So we've built this up over the hundreds of years of scientific inquiry, so that I may be telling you nothing that isn't just what you consider, oh, this is just doing good science. In fact, one of my dearest colleagues took a look at this formulation of what metrology is for the first time, and he says, well, that's just doing good science. And I'm like, yeah, that's a full sentence. Can I quote you on that? So what we've done is try to formalize methods to compare results, and that we typically term that metrological traceability. I show this as a three-legged stool. So there are three elements that we systematize. Traceability, metrological traceability, which is really, how did I get the units on this answer? Or how can I compare this answer, this result, to other results? Measurement uncertainty, which is, okay, I've got 37 of this and 42 of that. Are they the same or are they different? So what measurement uncertainty is it's really a reliability interval. It's a numeric quantity typically that describes the dispersion around that measurement result that can be asserted. Typically, you might be used to doing t-tests, you may be used to calculating confidence intervals. Those are the kinds of things that you need to systematically assess to quantitatively estimate measurement uncertainty. And method validation, which is a sort of horse of a different color. What method validation is what are the systematic ways I can use to test whether or not I'm seeing artifact, or whether or not what I've just reported is due only to the phenomenon I've just characterized and reported, or whether I'm starting to see, gee, on a rainy day, the needle on this measurement seems to be higher all the time, even when I'm measuring the same thing. In other words, I'm measuring barometric pressure, perhaps, or an artifact thereof. So how do you systematically do these things? And there you go. It supports the confidence in those measurement results and supports interoperability. So I can go to Paris and eat brie and baguettes until my cholesterol rises and hits the bell, and then I can fly home. And I know that the bell that it hit at, whatever it is, 240 if you want, is meaningful, because when I get home, it's still 240, and then 3 weeks later, it's down to 175 again. So in other words, how do I get this kind of comparability of measurement results across space and time, and know that that's meaningful? And so much of our science depends, frankly, the enterprise of doing science depends on having that in place, that kind of interoperability, so that I can go and take someone else's results and build on them to do the next advance, as everybody in the room is working towards. So this breaks when you try and sequence a genome and say, is this the same genome? Do I get the same answers as the lab down the street? Do I, if I'm measuring a gene expression signature and there's 47,000 transcripts that I'm reporting a value for, can I actually say that that 47,000 transcript signature that I measured today in my lab is comparable to the one I want to measure in 6 weeks or 6 months on the same series of samples in a longitudinal study? How do I establish that kind of comparability? And it's not through the traditional traceability, measurement, uncertainty, and validation efforts. We're having to invent some new approaches. And so that's the organizing principle of the group that I've built in a nutshell. And we've been at this for about a decade now. And what is the new measurement science? What do we have to figure out how to do? How do we use reference materials? I can't just tell you, oh, calibrate with this, and you'll get comparable results. How do I put out my data? How do I annotate my data so that someone else can actually look at the data and say, oh, I know what they did and I can reproduce that? It's much more complicated than when I'm measuring a single parameter or even tens of parameters, as is common in the analytical chemistry kinds of experiments. And really, the focus has been on the omics, and largely because it's easy and I'm lazy, we've been focusing on the genome and gene expression products. And as I say here, there's too many things to calibrate, too many things to use, too many things I am measuring to use the traditional methods. And many of these properties are identities. When I'm sequencing a genome, I'm measuring the order of the nucleotides. I'm not going to get into epigenetics and variants of that sort, but if I've got A, C, G, and T, and I want to tell you how confident I am, how do I do that? We don't have formulations in metrology that are good for that. I can't tell you it's A plus or minus G. That's sort of not a logical sequitur. And so we're trying to develop ways of systematically addressing confidence and building confidence. Again, so you get that confidence in your measurement result, you get that interoperability, and we can build on each other's work, scale what we're doing so that, as my colleague Drew Endy would put it, we can build the iPhone out of grass clippings using wood fungus that are highly engineered to build iPhones. Give that a few years, but that may be your iPhone 36, which there may be one someday. My group has typically worked because, again, I will note, I'm usually pretty lazy. We work with our customers so that I try and not so much get them to do the work, but get them to keep me from doing the stuff that I shouldn't bother with. So we form consortia, public-private, academic partnerships. We work with other federal agencies a lot. In this space, in the biomedical space, there's a lot of interest in working collaboratively with our partners at NIH, who have such an enormous investment portfolio that enriching the value of that portfolio is only sensible by having good measurements and reliable, reproducible results. And we've all had our ears to the radios or TVs or YouTube or whatever your common, watching reproducibility scandals happen on Twitter or looking at Retraction Watch or whatever it is we're doing, we're all aware of that. And I'm also going to give a disclaimer that I have a vested interest in measurement science solving these problems and metrology solving these problems, but I really believe that a lot of the reproducibility problem in biology has been that we don't know what we're measuring, or we're measuring the wrong thing, or our measurements are in fact artifacts, and that the phenomenon we're purporting and reporting and using to support or refute our hypotheses are in fact invisible to us or are masked with the noise of our measurements. And so watch this space. I think things are going to get better, and that's part of what gets me on the next airplane and up in the morning, et cetera. And the other partner agency that I need to throw in here is the FDA over in Silver Spring, who are really looking at how do you take the new technologies and bring them into regulated clinical practice. And I'm going to finish my talk this afternoon with a glimmer of what we're thinking about and doing there. So a suite of tools. We make materials, actually put stuff in tubes and sell that for cost recovery, so we really are losing money on every unit sale, and put that in your hands so that you can evaluate your processes, so you can calibrate your instruments, so you can do the three-legged stool of how much should I trust this? You can have the tools to do that. We do the same thing with data, making available reference data, which is evaluated and critically considered so that when you get those data, you understand where there's artifact or not. How much can I rely on that? We host and participate in interlab studies to benchmark labs against each other, or look at the population of performance within a community. Super informative. Wow, brand A always gives me this population of results, and brand B gives me this one. They may both be wrong. They probably are both wrong, but they don't agree. And then how do you harmonize, and where do you get to that? And so that's another tool in our kit. And we try and, as I say, do this methods development so that we can establish what are the principles for metrology at the genome scale. I'm going to give an overview of a project of ours that has been along these lines, that has been pretty well adopted and accepted in the early stages. I would count this a big success for us. And that's standards for the human genome. And so what it is it's human genomic DNA in a tube. We call it Genome in a Bottle. And we're doing that as a consortium with on the order of 100, but we're not really keeping count because it's a loose-knit consortium where nobody's signing papers to join, but on the order of 100 partners across the planet and across academic, public, and private sectors. I'm not going to talk about some other things we've been doing largely in the transcriptome, and I know there may be some interest in that, and I'm around to chat at breaks, but we put out a set of reference samples that underpin the external RNA controls consortium products. And you might have used some of these RNA spike-ins in your transcriptome work. And we've got a nice little R package that actually takes that and gives you some quality metrics and performance metrics for your work. And we've got some new methods using mixtures. And a colleague of mine is here in the room, Nate Olson, who's a NIST staffer who is in a PhD program here, who is trying to bring some of the technical approaches we've developed here into some of the research. So find Nate at breaks too. He's there. Looks like he walked through the rain to get here. So there's a quick ad for this paper that talks about evaluating transcriptome measurement performance. Nature Communications back in 2014. Sarah Monroe was first author. And a quick ad on the mixture design stuff, and this is Jared Parsons' first author, and this was just published in September, I think, of this year, and it's using these mixture designs to evaluate whole transcriptome RNA-seq measurements, and basically giving you your answer and a target, and that paper is BMC Genomics. But I want to zoom in to this Genome in a Bottle project because it's been impactful already, and it turns out that we were timely in initiating this endeavor. In January of this year, January of 15, President Obama made a signature call for a precision medicine initiative, and I know we'll hear more about that later this afternoon. You don't always get to see the president in the east wing of the White House standing next to a model of a double helix DNA. And the backstory on this is that that model, the people from the Office of Science and Technology Policy were visiting Eric Green at the NHGRI, and that model was standing outside of Eric's office. And they were like, can we take this? And so that model went from Eric's office, outer office, to the White House for this event. That's been a popular picture. We introduced in May of this year the first Genome in a Bottle reference material product, and that is whole human genomic DNA characterized for its variants, and I will get into more detail on what that means. Made that available as a reference material from NIST, and the New York Times featured it on it. And really, this is my tagline, not the New York Times headline, but so you've sequenced my genome, how well did you do? What does that mean to say that we've sequenced a genome? And how do you use those data is yet another story. So what we're trying to do is we're trying to release a product that takes, and this is some generic genome sequencing, what I would call a measurement process, where you start with a tissue sample of some sort, blood. Many people have probably spit into a tube and sent off their saliva sample for a genotyping assay at 23andMe or Navigenics. Navigenics is probably gone by now, but Ancestry.com. But you take that sample, you do some genomic DNA isolation, then you put it into this sequencing process, which for next-gen sequencing looks a lot like making a library of sequenceable targets and doing the sequencing, and then figuring out what you've done in making this library is shredded the genome. So if your genome is 23 chromosomes and they're on the order of 10 megabases to 100 megabases long, we're not sequencing your genome. We're sequencing little pieces that we've generated as a fragment. So we have to put it back together bioinformatically. Then we want to say, gee, we've put this estimate of what the genome is back together. How does this differ from what we would consider, and I hesitate to use this word, but what we would consider normal? What we're looking for typically when we are sequencing a genome and trying to use it in a clinical setting is pathology. And so how do we infer pathology from the genome to the very poor degree that we can do that? And we'll hear more about that today too. But we do this process called variant calling and looking at the differences from the genome that we've proposed is the best solution for these fragments that think we've sequenced. And then we come up with some kind of confidence estimation, and then there's a downstream clinical analysis. So these genome in a bottle samples, what are they? They're the traffic light. And here's the secret. This kind of traffic light sample is used, if we go back to that three-legged stool, as a validation tool. Really what we're doing is giving you evidence to say that this experiment happened well or not. So that doesn't give you a green light from your traffic light. That gives you your proceed with caution. It's a yellow light, or flashing yellow light. I guess you should brake and stop if it's a yellow light. I'm learning that now. So proceed with caution, and then start trying to test your hypotheses. And we've got a handful of materials. And why did we get into this? This is a beautiful Venn diagram, which is not the most current Venn diagram, but it's the prettiest Venn diagram. And what this shows is the agreement or disagreement when a number of different techniques, and these in this case were a single technology, but multiple bioinformatics processes, look at the same sample. And this is for variant calling for small, really single nucleotide variants in this case. And what you see is if you bring enough players into the game is you've got on the order of 60% in agreement, where everybody says, yes, this is that. And so you've sequenced my genome, how well did you do? If somebody else takes a look at it, they're going to get a different answer. And who's right? Is anyone right? And obviously nobody's right, but who's the rightest? Or how can we upgrade everybody to be righter? So we spun up this consortium because we didn't have all the answers either, and are developing a portfolio of samples. Critically, we're using really well-consented samples, and this is important for our product development. So these samples end up being commercially available. Even before we are distributing the samples, our colleagues and customers are already using them. We're making available high-accuracy reference data for, again, as a collaborative project, and tools to facilitate their use, and we're working with a different project, the Global Alliance for Genomics and Health, to develop these benchmarking tools. So our customers can ultimately measure one of our samples and ask how well they did when benchmarked against our reference data. Let me zoom through that animation. Importantly to note, we picked the first sample from a large family that had been used since the 1980s in lots of genetics and genomics studies. And this is because it's a sample that people are familiar with and have already been measuring in their labs. This is now the first benchmark sample out there. And it's the mother of a 3-generation, 17-member family. And so she has 11 progeny or offspring, and we have genomes for her husband, her husband's parents, her parents, and all 11 of the children. We are doing 2 new trios, an Ashkenazim Jewish trio and a Han Chinese trio that were from this personal genome project up in, that's operated out of Harvard that has that state-of-the-art consent. So how does this look and how do these get used? Back to our measurement process, there's sort of a pre-analytical, an analytical, and clinical process. That those yellow boxes are this analytical process that we're testing with. Our sample goes in the top, and you get out, this is an animation that my colleague Steve Lincoln from Invitae did. So we can thank Steve for the Mickey Mouse wizard there. That's where his focus has been, is trying to figure out how do I compare. And our data, our reference data, critically match up with the material and go into that comparator. We've got a nice paper out in 2014 on how do we come up with our reference data, because who checks the checkers? And so what we did was we came up with essentially a machine learning arbitration process to take data from multiple methods that were both different sequencing methods and different bioinformatics methods, and not just vote, 3 said yes and 3 said no, or 2 said no, but actually use all the inferential data around the measurement to know how confident we should be in each of them, and then use an informed decision-making process to do our variant calling. And so what we came up with, because I've talked a little bit about measurement uncertainty, there are millions of variants in the genome, that in that case not a phased genome, so the sort of 3.2 gigabase genome, millions and millions of variants, on the order of 4.5 million variants. There's no way to put a measurement uncertainty on each of them. And so we came up, even a probabilistic estimate, say I'm 80% sure in this one and 76% sure in that. We came up with essentially a 2-level rating, which is we're either confident or we're not confident. And we did not report genotypes or variant calls where we weren't confident. And so we did about 77% of the genome. And there's 23% of the genome that we're essentially blind to. And these are regions that are difficult and variant types that are difficult, or easy variants in a complex variant embedded within. And so we're making progress, and this morning Hector asked me, so what's next for Genome in a Bottle? And it's getting to more difficult variants in more difficult regions. And I'm not considering that we're going to be done anytime really soon, because as soon as we get to the next kind of variants, structural variants in difficult regions still remains ahead of us, and then difficult regions become more difficult. And then we can start looking at mosaicism within a genome or within an organism. So we've got pretty strong rule sets for adjudicating this stuff. And we're using the heritability information. Lee first reported on mutation rates in the family of 4, that quad, I don't remember the journal, but back in 2009, I think, Lee. Science in 2009. And so Lee demonstrated well ahead of us that you can use this Mendelian heritability to benchmark not only the measurements, but to estimate the basic mutation rates with heritability. And we're using that kind of approach also to validate our metrology of the genome. We've got these new trios that we're in the process of packaging up as reference materials and getting out the door. Lots of sequence data. That table's not really meant to be parsed so much as what we've done is taken the state-of-the-art in sequencing approaches, and we're trying to integrate that into a best estimate of the variant calls for these new samples, these new benchmark samples. These data are described in this bioRxiv preprint, and that's the paper's currently under review at Nature Scientific Data. This Genome in a Bottle organization has biannual workshops, and we do some episodic telecons to coordinate our processing. And we wrote up our August workshop as a description and commentary in Cell Systems. Came out in September. Lots of people using these data, and it's exciting because these people were using these data before we released the reference material even. It's the advantage of using a widely available sample. We're using whatever tools we can to get things out. Partners at NCBI and NIH, National Library of Medicine, are helping us make these data available. So these data are available to you right now on your laptops in this room to start playing with through our GitHub site. As I said, we're working on benchmarking, working really hard on benchmarking and doing that in partnership with the Global Alliance, which is also the science fiction-iest name. I'm excited for the new Hunger Games movie, and I'm sure the Global Alliance will be featured in the 5th one, or 6th one, I guess we're getting. Anyway, but what we're trying to do systematically is bring the principles of metrology to the genome. I'm going to check my time. I've got a couple more minutes, so I'm gonna go through one more set of interesting ways forward on building on this genome in a bottle. One note I said earlier, that's important that we're selecting these samples to be commercially reusable, consented with this PGP consent. Again, beating us to the punch, before the NIST reference material is available, there are commercial reference materials or reference samples available from 2 different companies, Acrometrics and Horizon Diagnostics, and they're trying to bring these samples in to practical clinical process controls ahead of the game. It totally, for me, validated my decision to use genomes that were not yet characterized but were consented appropriately for these downstream uses. That consent also covers making IPS lines and differentiating those IPS lines and commercializing those. The individuals who are the donors of these genomes are consented to make themselves known, and and have done so. I'm going to leave it at that. And so we're sort of pushing the edge. What does it mean to be a reference material genome? So I also talked a little bit about what does it take to get these into clinical regulated, what does it take to get genome sequencing into clinical regulated applications? There's a lot of folks buzzing around the beehive of this question. FDA had 2 workshops last week, one on analytical validation and one on using clinical databases for clinical interpretation of genomes. There's a couple of quotes I've got up here. We're working closely with our partners at FDA to understand what the needs are and how to do this. And here's where I put on a different academic hat. I'm a faculty on the Center of Excellence in Regulatory Science and Innovation that is hosted at UCSF and Stanford. And in my work there, as well as in my work with our partners at FDA, federal partners, we've been looking at how do you build a regulatory system to assure safety and efficacy of genome sequencing in clinical applications. And we do our typical thing of breaking it down into a measurement process and looking at the pieces of that. So what's a standards architecture to support the safety and efficacy? And you have to chunk it because you're not going to do it all in one big fell swoop. And so we take our approach. This is not dissimilar from what you've seen on the genome in a bottle. This is the analytical performance piece that I showed you in the yellow boxes. And this is a work that's been accepted for publication, so I'm showing it here now. With that proviso, we built a big table that basically identifies the research agenda that the CERCs, these Centers of Excellence in Regulatory Science and Innovation, and the academic investigators can start pursuing to develop the appropriate standards to make this happen. And again, we're doing this in partnership with, we're NIST and Department of Commerce, and in partnership with our commercial industrial partners. So I've broken it down into sort of what evidence do you need to establish at this stage of the game? What are the practices that you can use to establish that? What standards are needed? Who cares? Who are the right stakeholders here? You know, at one point it's the clinical labs, at another point it's the professional societies, at another point it's the FDA themselves. And what are the knowledge gaps? Because the knowledge gaps can be used to identify that research agenda. And so I'm not going to belabor you with this except to say that for each stage we've got a table that shows some of the examples. And that's the direction we're taking the metrology of genome-scale measurements. You know, how can I be confident, and what infrastructure can we build to make that happen? And as I say, I'm not going to take the time to do that. I want to acknowledge my partners at NIST, my partners in the Genome in a Bottle Consortium, the GA4GH benchmarking team, who are doing some super innovative work in some really difficult information management and comparing variants as non-trivial or variant calls. My partners at Stanford and UCSF and FDA, who have been awesome to work with, as well as been great funders of the work in my group over the past 5 years or so. So thank you so much for your engagement, and I'm looking forward to the rest of the afternoon with you. And we have time for questions, I think.
Time for questions for Dr. Salit?
It's just after lunch, so. And thank you. Do we have one? Huh? Please. I'm sorry, I missed, can you go to the mic maybe? Sorry, there's a fan over my head and my ears are getting old.
Do you think that the next step will to basically target specific maybe diseases to identify faster and maybe even change the genome?
I'm going to forward reference you to Lee's talk at 5 o'clock this afternoon, but lots of people are totally trying to do that, to use the genome to either predict disease or use, for instance, in cancer, which is a disease that has lots of associated genomic variants, using the genomic variants to try and do therapeutic decision-making, treatment, so diagnosis. And so yes, I do think that we'll see more and more investment and successes and some disappointments in using the genome, especially as a static entity, to try and predict and diagnose disease. But again, I think Lee's going to have a fun treatment of that later. So an ad for the Lee.
Seems to me one of the challenges in genome assembly is identifying insertions and deletions of all sizes and everything. So if you've done any comparative analysis comparing the short read technologies against longer read technologies, PacBio, Oxford Nanopore, to look at that whole, it seems to me once we get
So I'm going to, yes is the answer. We are looking at that, and we've got some preliminary results. I don't have a slide of those results, but the long-read data are working the way you think they would. And this list of sequencing platforms, data sources for the Ashkenazim Jewish trio, one of the things we did is we invested in quite a bit in the, and is it up there? The PacBio long reads to the degree of about 70x on the son and 30x on each of the parents. And there are a number of groups independently working on assemblies of those. And the answers are, as you would expect, much more informative. And then the read cloud or pseudo long read work from 10x Genomics and long fragment reads from Complete and the molecular work are contributing to that too. And we have not completed the analysis. These data are what we're publishing in that Nature Scientific Data paper. And gosh, we should have recommended you as a reviewer. But that's the right question, and you're aligned with our thinking on it, is that we'll not only insertions and deletions, but rearrangements of all sorts. I don't have a crisp study. I would point to some of the studies that compare DNA microarray measurements to RNA-seq measurements and compare platform to platform. And when you pick through where you find discrepancies, when you pick through those discrepancies, we can usually identify an analytical failure that gives rise to them. You can find why you're getting the wrong answer. And we can talk more offline on that.
So I was curious, you're showing the genomic measurement standards, but in the clinic you also have clinical images and other biomedical data. What are the differences and similarities in your work?
And I'm a chemist, so, and this is a cappella. Looking at the imaging data, I think the kinds of artifacts that we're prone to see is what you would expect from image analysis artifacts, as well as interferences from your imaging modality. So frankly, I think we would just draw a different schematic or flow diagram of the measurement process and then identify throughout. So the approaches I would take would be largely similar. And I think some of the challenges are not all that different. I don't know if Elizabeth's going to show some imaging, some cell imaging, but you'll see some of what I'm talking about, I think, in the next talk. Thank you guys again.